Hello class. Next we will discuss plant respiration. We will explain from three aspects: the concept and classification of respiration, respiratory metabolic pathways, and the respiratory chain and electron transfer. The first content, the concept and classification of respiration. 1. The concept of plant respiration. Respiration refers to the process in which organic matter within living cells, with the participation of enzymes, is gradually oxidized and decomposed, releasing energy. 2. Classification of plant respiration. 1. Aerobic respiration refers to the process in which living cells use O2 to thoroughly oxidize and decompose organic matter, releasing CO2 and H2O, while also releasing energy. 2. Anaerobic respiration, also known as fermentation. It refers to the process in which living cells carry out incomplete oxidation and decomposition of organic matter in the absence of oxygen, releasing some energy at the same time. Anaerobic respiration is divided into two types: alcohol fermentation and lactic acid fermentation. In the chemical equation, we can see that aerobic respiration produces more energy than anaerobic respiration. The second content, respiratory metabolic pathways of plants. The material changes of respiration are manifested in the gradual oxidative degradation of the respiratory substrate. The degradation of polysaccharides, fats, proteins, and other biological macromolecules is centered on the degradation of monosaccharide molecules, including glycolysis, the tricarboxylic acid cycle, and the pentose phosphate pathway, among other metabolic pathways. 1. Glycolysis. 1. Definition. The process of hexose decomposition into pyruvate in the cytoplasm is called glycolysis, also known as the EMP pathway. 2. The reaction site of glycolysis is in the cytoplasm. 3. The total reaction formula of glycolysis is as follows. For glycolysis includes a total of 10 steps, which can be divided into 3 stages. The first stage, hexose activation stage including steps, and its essence is the consumption of ATP to produce hexose phosphate esters. The second stage, the cleavage reaction stage, including steps and, first fructose 1,6-diphosphate is cleaved into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The two 3-carbon sugars can interconvert under the catalysis of isomerase, so it can be regarded as the conversion of fructose 1,6-diphosphate into 2 molecules of glyceraldehyde 3-phosphate. The third stage, the oxidation and energy production stage, including steps 2, glyceraldehyde 3-phosphate undergoes oxidation, producing reduced coenzyme INADH. ATP is generated in step 5. The significance of glycolysis. Glycolysis is a common pathway for both aerobic and anaerobic respiration in higher plants. Pyruvate, the product of glycolysis, is further decomposed into ethanol or lactic acid in the absence of oxygen, or enters the mitochondria under aerobic conditions, where it undergoes gradual decarboxylation and dehydrogenation, complete oxidative decomposition, forming water and carbon dioxide and releasing energy. The subsequent aerobic process we are going to discuss is the tricarboxylic acid cycle. 2. The tricarboxylic acid cycle. Tricarboxylic acid cycle, abbreviated as TCA cycle, also known as the citric acid cycle and the Krebs cycle. The tricarboxylic acid cycle takes place within mitochondria. 1. Chemical process of the tricarboxylic acid cycle. There are 9 steps in the reaction which can be divided into 2 stages. 1. Preparation stage, oxidative decarboxylation of pyruvate. Under aerobic conditions, pyruvate enters the mitochondria and generates acetyl-CoA and NADH through oxidative decarboxylation. The acetyl-CoA then enters the tricarboxylic acid cycle for complete decomposition. The oxidative decarboxylation of pyruvate is the bridge between glycolysis and the tricarboxylic acid cycle. 2. The oxidation of acetyl-CoA in the tricarboxylic acid cycle can be divided into 3 stages. Formation of citric acid, acetyl-CoA plus oxaloacetate citrate. Oxidative decarboxylation, including 4 reactions, namely the formation of isocitrate, oxidative decarboxylation of isocitrate, oxidative decarboxylation of alpha-ketoglutarate, and the generation of succinate. This stage releases 2 molecules of CO2 and synthesizes ATP. Regeneration of oxaloacetate, including 3 reactions, succinate goes through the generation of fumarate and malate, and finally to oxaloacetate. As can be seen from the figure, pyruvate first undergoes decarboxylation and dehydrogenation to form acetyl-CoA, then enters the tricarboxylic acid cycle, and continues to undergo decarboxylation and dehydrogenation within the cycle, until it is completely oxidized and decomposed. For every molecule of pyruvate broken down, 3 molecules of CO2 are released. Both the 5 pairs of hydrogen released by pyruvate oxidation and the pair of hydrogen produced by glycolysis are transferred to the respiratory chain in the form of NADH, or FADH2. These then release energy during electron transport, producing a large amount of ATP, and finally combine with oxygen to form water. 2. Physiological significance of the tricarboxylic acid cycle. 1. The TCA cycle is the main source of energy required for life activities. The 8-NADH and 2-FADH2 produced from one molecule of glucose can undergo oxidative phosphorylation coupled with the respiratory chain to produce a large amount of ATP, which provides energy for plant life. 2. The tricarboxylic acid cycle is the hub of substance metabolism. This cycle is the common pathway for the complete oxidation and decomposition of sugars, fats, and proteins. It can also be connected and transformed with other metabolic pathways through metabolic intermediates. 3. Other metabolic pathways. 1. Pentose phosphate pathway. 2. The pentose phosphate pathway refers to an aerobic respiration pathway, in which glucose is directly oxidized and decarboxylated in the cytoplasm, with pentose phosphates as important intermediate products. 3. Glyoxylate cycle. During germination, the stored fatty acids are decomposed via beta-oxidation into acetyl-CoA. This acetyl-CoA is transformed in the glyoxylate cycle body through a cyclic enzymatic reaction process that produces glyoxylate. This process is known as the glyoxylate cycle. The third content. Respiratory chain and electron transport. The NADH plus H plus and FADH2 produced in glycolysis and the tricarboxylic acid cycle cannot directly combine with free oxygen molecules and need to be transferred through the electron transport chain before they can combine with oxygen. The respiratory chain, also known as the electron transport chain, is an orbit composed of multiple electron carriers located in the inner membrane of mitochondria, arranged in a certain order to transfer electrons to molecular oxygen. The carriers of the electron transport chain can be divided into hydrogen carriers and electron carriers. Hydrogen carriers, including the cofactors of dehydrogenases, mainly NAD+, flavin mononucleotide FMN, and flavin adenine dinucleotide FAD, coenzyme Q, CoQ, and so on. They can transfer both electrons and protons. Electron carriers, including the cytochrome system and certain flavoproteins and iron-sulfur proteins, which transfer only electrons. These carriers form 2 respiratory chains. The first, called the NADH respiratory chain. The second, called the FADH2 respiratory chain. That's all for today. Thank you.